All right, let's dive into something pretty incredible. We're going to decode a secret language, one that's being spoken every single second inside your body. It's this silent, super high-stakes conversation between your immune cells, and it's the network that literally keeps you alive. So how does it all work?
I mean, really think about that for a second. Your body is this bustling city of trillions of cells. So how does a single immune cell in, say, your bloodstream let another cell way over in your big toe know that there's a problem? How on earth do they coordinate a defense against an invader they can't even see? It's a massive, massive challenge of communication.
And that really is the heart of the issue. Your immune cells, they're scattered everywhere, patrolling every nook and cranny of your body. They have to find these tiny microscopic threats. We're talking viruses, bacteria. And then they have to communicate with each other over huge distances to rally a defense. You know, it's kind of like trying to organize a citywide emergency response where everyone can only whisper.
So how in the world do they pull it off? Well, it all starts with something that's actually beautiful in its simplicity, a molecular handshake, a real physical interaction between two molecules that lets one cell pass a message to another. This handshake? It's the basic alphabet of the immune system's secret language.
Okay, so this handshake has two main players. On the surface of one cell, you have a receptor. Just think of it like a tiny antenna that's perfectly shaped to pick up one and only one type of signal. And that signal, it's a molecule we call a ligand. When the right ligand, the right molecular message, bumps into its matching antenna, they lock together. And just like that, the message is delivered. But here's the thing. Not all handshakes are created equal. The stickiness of this connection is called affinity. A high affinity bond, that's like a firm, confident handshake. The message is loud and clear. A low affinity bond, on the other hand, is more like a quick, fleeting brush of the fingers. The connection is weak, and the signal might not get through at all. So the strength of this grip, it determines absolutely everything.
And this is where things get just incredible. Our immune system can actually learn. At the beginning of an infection, an antibody might have a pretty weak, kind of clumsy grip on an invader. But over time, our body refines these antibodies, making them stickier and stickier and stickier. By how much? A staggering 10 million times stickier. It is literally evolution happening in real time inside your own body.
Okay, so a single strong handshake is great. But what if you could have a bunch of them all at once? Well, that's the next level of immune communication. It's the difference between a simple handshake and a full-on, you are not getting away hug. And this image right here shows you exactly why that's so powerful. Look at that single connection up top. It can break apart pretty easily, right? The signal just kind of flickers on and off. But now, look at the one on the bottom. When a molecule has two binding arms, it can grab on in two places at once. So even if one arm lets go for a second, the other one holds on tight, keeping that signal locked in.
This amazing collective strength that you get from multiple bonds, it has a special name, avidity. And it is a core principle of how we stay healthy. By taking a bunch of individually weak connections and putting them all together, the immune system creates this incredibly strong and specific overall bond. It makes sure that when a cell finds its target, it doesn't just let go by mistake. Now, the molecule that is the absolute superstar of all these principles is the antibody. But figuring out its elegant structure, that was not easy. It was this amazing scientific detective story that took decades to solve, with scientists uncovering its secrets one clue at a time.
So this story kicks off all the way back in 1939. That's when scientists first managed to track down antibodies to a specific part of our blood they called the gamma globulins. It was a huge first step, but what the molecule actually looked like was a total mystery. It took more than 30 years, all the way until 1972, for Rodney Porter and Gerald Edelman to finally figure out that iconic Y shape we know today, and yep, that work earned them a Nobel Prize.
The first major clue came from this just beautifully simple experiment. So, scientists took blood serum from a rabbit that had been immunized. That's the blue line you see, with all its different protein peaks. Then, they simply mixed that serum with the target, the antigen, which made all the antibodies clump up and fall out. And when they reran the sample, look at that black line. The gamma globulin peak, it just disappears. Poof! They had found where the antibodies were hiding.
The next huge breakthrough came from using enzymes as a kind of molecular scissors to carefully snip the antibody into pieces. And as you can see here, one enzyme, papin, cut the Y into 3 bits. 2 of those bits, which they called Fab fragments, were these identical arms that could still grab onto a target. The third piece was the base, the FC fragment. This was the smoking gun. It proved without a doubt that every antibody has two identical binding sites, the perfect design for that powerful two-handed grip of high avidity.
And if we zoom all the way in, right to the very tip of the antibody's arms, we find the business end. The part that actually does the grabbing is made of these incredibly flexible and variable protein loops. You can see them here in red. They're called complementarity determining regions, or CDRs. By just changing the shape of these loops, our body can create billions of different antibodies, each one tailor-made to grab onto a completely unique molecular shape. It's incredible.
Now, this amazing specificity is the signature of what we call the adaptive immune system. But it's not our only line of defense, not by a long shot. Our body actually has a two-tiered security system, and each one has a totally different strategy for spotting trouble.
First up is the innate system. Think of this as the security guards on patrol. It uses a small toolkit, only about 100 types of receptors that we inherit, to look for really broad, general patterns that are common to lots of microbes. It's super fast, it acts immediately. Then you have the adaptive system, with its T cells and B cells. These are the special forces. It takes them about 7 to 10 days to really get going, but it can generate billions of unique receptors by shuffling its own DNA. This allows it to recognize and, most importantly, remember almost any specific enemy it ever meets.
So why does understanding all this stuff, this secret language, why does it matter so much? Because by learning to read this molecular conversation, we've essentially learned how to write the blueprint for a huge chunk of modern medicine.
I mean, really, this fundamental science is behind some of our greatest medical achievements. It's how we design vaccines that properly train our adaptive system. It's how we create drugs like TNF-alpha blockers for arthritis that can tell a harmful immune conversation to quiet down. It's the entire basis for revolutionary cancer therapies that unleash our own immune system against tumors. And it lets us treat devastating immunodeficiencies because we can figure out exactly where the communication is breaking down. So we've spent the better part of a century learning to decode this unbelievably complex language of receptors and ligands, affinity and avidity. We've gotten really good at listening in on the conversations that keep us healthy. The question now is, what's next? As we move from just reading the messages to maybe, just maybe, starting to write our own, what incredible new possibilities are we going to unlock for our health?